Good morning, everyone. So I'm Puriya, and in this part of the course, Bio 390, my aim actually is to walk you through some of the interesting ideas in network theory and the relevance in biology, basically. So we are biologists, so we are interested to see how we can use theories from network, mathematics of networks, basically, and some other ideas in that we actually can borrow from network theory to better understand biological systems. I'm a postdoc at the University of Zurich. At the same time, I have this affiliation with Harvard. And my research is in evolution. So I'm interested to ask questions regarding the evolution of the systems. And then quite often we come across ideas from network theory, and then we try to understand it from those perspectives. So as I said, we try to have this course or this lecture in two parts. So there would be 45 minutes. I guess we started a bit late, so maybe we go fast in the beginning. And we have 15 minutes break, and then the second part. So in the first part, I'm going to talk about examples of networks. And in the second session, I'm going to talk about properties of networks. Yes, so as I said, this is an interactive course. I really don't like the idea of talking and then you keeping notes and then basically coming for the exam. So I thought what would be really interesting is really ask questions and then you answer those questions. And all the wrong answers are welcome as well. So the first question for you is, what is a network? Oh, I thought you want to answer. Anyone, just, shoot. You have coffee problem? Oh, please go ahead.
So network is like there are several components interacting with each other or then in specific order to each other.
Perfect. That's a good answer. Anyone wants to object with this opinion? Or maybe complement it?
I would expand on it, saying that it's not just interaction per se, there might be some other sort of connection.
Yes, that is true. That is good. And both answers are correct. I think this is actually the answer to the question. So if we follow this way, I guess the course would be very simple, so right answers. Basically, a network is a collection or a set of components or entities that can interact. People are talking also about networks that have either direct or indirect interactions, but in principle, when we have more than two components or elements or entities, and these guys can interact with each other, we can think of having a network. And then we can think about having structures and having a sort of topology of these networks. How do they arrange in the space? How do they interact with each other? And so on and so forth. When we're talking about biological networks, we are talking about networks that encompass different biological entities, such as DNA, protein, cell, organisms, humans, social networks. These are all biological networks, basically. So as long as those nodes and those entities have biological meaning, right? So either they're molecules or they're organisms, we can define those networks as biological networks. The first order of business here is that we have molecules and structures. And those structures, those macromolecules basically form a network. But basically, the right arrangement of these molecules and then the sort of interaction that they have with each other is more of a chemical structure or chemical network. So on my laptop, I prepared very interesting PyMol sessions when you could go through some of these proteins, but I don't have the luxury today. So I just want to ask the question from you that we can look at the protein from two perspectives. So one is that this is a macromolecule. This is a set of interacting atoms. And the other one is a view that is more biological. Anyone has any idea on that? Let's say I want to define two networks. One is chemical, of course, because I can say that there is an atom here, and this specific atom is connected to the other atom, but these are amino acids, not atoms. But they are composed of different atoms. So one view, of course, is the chemical structure view of it. So the other view is the biological view of a protein. How we can think of a network in protein, please?
Maybe the 3D shapes or the alpha helix and the position.
Perfect. That's the right answer. So 3D structure of the protein constitutes another layer of biological information. And often it's something that we care because it actually linked to what we call the second dogma of molecular biology. What's the first dogma? We all know that, right? So DNA, RNA, and protein. The second dogma is basically more centered on proteins. So we say sequence, structure, function. Of course, this dogma is challenged by the presence of proteins that have disordered structures, regions like this. But in general, most proteins have structures, and those structures help us better understand their function in the cell. Yeah, this is the question that I asked, and your friend answered very well. Then, what we have on top of proteins, or on top of these molecules, is the network of protein-protein interactions. So these proteins can interact as well. Again, I've prepared this PyMol session that you can go and see different components of these proteins. But when I'm talking about interactions, these are really physical interactions, let's say hydrogen bonds or electrostatic interactions. And proteins can interact with each other. That's why we get a network of protein-protein interactions. And these networks are very important. And often what we see is that a bunch of proteins would come together to form a complex. And many cellular processes in the cell are achieved by complexes. For example, we know that men has X and Y complex, X and Y chromosome. Females have two X chromosomes. What happens is that one of these X chromosomes need to be silenced. So the whole process of silencing a chromosome is orchestrated by a huge complex having 10 or more components. So they gather and they sit on the chromosome and they make sure that one of the X chromosomes would not be activated. So this is another layer and very interesting point because we can already try to understand and get the complexity of these systems. Okay, so I guess most of you have laptops. What is really interesting here and what's really important for me at least is to try to see where do we get this information from. How many of you are familiar with the string database? Okay, that's a good sign. So we can actually play a little bit with that. So let's spend some minutes on this data set. So it's called a string data set. So you just need to put a string in Google, and then you get this data set. What you see in this data set is the presence of about 14,000 organisms, 67 million proteins, and more than 20 billion interactions that exist between these proteins. So what I would like you to do, maybe for 5 or 10 minutes, is go through this database and search two proteins. So you can have two browsers. In the first one, you search for myoglobin. That's a protein that is in our muscle. The main job is to take oxygen from hemoglobin and delivers it to the muscle cells. And the second one is DNA-directed RNA polymerase II. So you can actually go to the database, choose your organism there, and then put these sort of acronyms, the gene names there. So let's spend some minutes on it. I'll try to see if I can use this computer to do the same job. So is anyone totally lost? No, that's good. So I assume you went through that. Do we need more time, or anybody is interested to talk about it, the observations? What do we see? Yeah, please go ahead.
The protein network of RTB1 is much more complex, like every single component is interacting with each other, whereas myoglobin has defined interactions, and not every single protein is interacting with each other protein network.
That's perfect. That's the right answer. And what we see here is that, of course, there are many colors, many edges with different colors. So these are interactions that are defined differently. Some of them are coming from physical interactions. Some of them are happening because of co-expression, as we get to it in the lecture. But when you compare the interacting network or interacting partners or interaction network of myoglobin with that of RNA polymerase II, we see there are so many interacting partners in this case. What's more interesting is if I try to play with the options that this database gives me. For example, I can actually go here to setting, and I can, so you see here, these are different sources of the interactions. It could come from text mining, meaning that this protein and the other protein were found in similar, corpus of studies or literature. But one interesting point here, because sometimes we're really interested in the proteins that physically interact, it could come from only experiments. So if I remove all the sticks and then only get this check mark here, which is the expression of, which is the experiment. So you see these are, and you need to update it, of course. Well, myoglobin becomes alone, basically. So we don't have many partners for myoglobin. But if we go to the case of POL2 and then repeat this exercise, you see there are still many components. The reason is that all these proteins are part of a complex. So they really need to assemble by different types of interactions, hydrogen bonding, electrostatics. You might actually come across these interactions in biochemistry, and they assemble the complex. So that's one example of a biological network that we're interested in. Please.
Do the distances between the nodes play a role in this network? Because the myoglobin seems to be more spread than the...
Not in this presentation, but distances between nodes could be quantified. For example, you could say that if, in some studies like quantitative genetics, when people are interested to see if two locus, if two parts of DNA are inherited together, the distance actually might be showed that way. But that's a good thought, because what it shows is that, I'll come to it in some of the slides, we don't only present graphs, we also do a bunch of other things on it. So we can represent the width of the edges, we can actually color them, we can do calculations, which I'll get to it. But good question. So let me ask this sort of fundamental question that I think might be interesting. So why do we have different tissues and cell types if we only have one DNA molecule? Any answer? Again, wrong answers are very welcome because it helps us discuss. So nothing is graded, just feel free. Say whatever you want. Well, that's a good question, right? Is anyone? Okay, good, go.
Different parts of the DNA are expressed in different cell types.
Good point. Good point. That's actually part of the answer. Anyone wants to complement it or complete it? Well, that's actually a good part of the answer. So the answer is the existence of another network in biology that we call gene regulatory networks. So these are networks of DNA and protein interactions. And these networks help sort of selective expression of markers and genes and other systems, basically. It's very complex, so that's why I actually generalize it. But the simple case here is that we have 3 genes. So these 3 genes have products such as mRNA and proteins. their proteins might regulate the expression of other genes, and we might have a feedback loop at some point. We might have cross interactions, and the end result is what we call gene regulatory network. So just simply the definition is that we have a set of molecular regulators that interact with each other to govern the gene expression of mRNA and proteins. And usually the gene expression program is what gives cells the function or the specificity that we are interested. For example, this is also very important in diseases. In cancer cells, we know that there are perturbations in this network. So what we expect to be the sort of gene regulatory network of a healthy cell now becomes cancerous, becomes different. Okay, so I'm going to ask another fundamental question. So what we know from, let's say, decades of experiments in biochemistry and cellular biology is that cells can cope with the stress. There are so many stress conditions that we might face: heat shock, chemical stress, starvations. We don't die immediately, after one day of not eating food, nor E. coli or yeast. So it's something that we have in common. So we can actually tolerate stress. And the stress is often achieved by a coordinated response of many genes. So how would such a coordinated response be programmed? So any idea? How do we get this sort of coordinated response?
Several genes that are all controlled by the same transcription factor.
That's the right answer. Perfect. So in general, what we have is gene co-expression network. So these are genes that could be controlled by the same transcription factor. And it's very interesting because there's basically a subtype of these networks might be stronger. So these genes, in principle, as you can see here, this is stick now. So these genes, in principle, can have some sort of coordinated response, all of them together, some baseline levels. But then maybe in some conditions, we have a selective module or a selective set of these genes that would show a very strong co-expression. And this is something that drives coordinated response, as I said. For example, one thing that we know is that when cells are stressed, transcription and translations go down dramatically. So we have ratios like 300-fold reduction in the transcription process. And this is often happening by that. One important thing to bear in mind is that we can quantify these networks. So as you can see here, we need experiments. That's one of the points that's really important for us, let's say bioinformaticians or people who are dealing with data sets. So often we need to deal, with or work with experiments. In this case, we have RNA-seq experiments or microarray experiments. I'm not going to really in the details of these experiments, but basically what we do here, we try to guess the expression of genes, and these are high-throughput methods, meaning that we can, in principle, look at expression of many genes, hundreds of genes. And then let's say we get the matrix like that. The rows of this matrix are our genes of interest, and the columns are samples that we have. So we can turn this matrix into a similarity matrix, which would show simply the Pearson correlation between these genes. So let's say we have gene number 1 and 2. We can see what's the correlation or what's actually the degree of covariation between these values and these values. And then we can put all these values here. Of course, what matters to us is the off-diagonal elements because it doesn't make sense to say a gene is co-regulated with itself. And we can deal with such a matrix when we have absolute values, or what we can do is we can put a threshold. So we can say as long as 2 genes vary and this covariation is more than 0.8, so we have a significant threshold, and then we put a 1 here. And these sort of binary matrices are really the most important elements that we can use for building networks. So as you can see, in this case, we can really relate a matrix to a network. So just look at it. So here we have node 1. We know that node 1 is being co-expressed by, gene 1 is being co-expressed by 9 and 7, so you can go all the way and see, yeah, that's the case. 1, 9, and 7, and 8 as well. So there are 3 1s here. So zeros, Zeros here mean that we don't have zeros means we don't have a co-expression. Ones mean that we have co-expression with other genes. One very, very important point here is that binary matrices and networks are always sort of equivalent. So we can build a network from a binary matrix. And it's very important to know that because sometimes you get a network, so the first question is that how can I do some calculations with this network? So what you do is that you try to convert this to a binary matrix and then do further calculations with that. Okay, so another class of networks that are really interesting are metabolic networks. So you might have heard about these networks. A metabolic network is a set of chemical reactions that produces either energy or building blocks for our cells, such as nucleotides or amino acids. And well, these reactions are catalyzed by enzymes. And what's really interesting about metabolic networks is that it's really proportional, or basically it's really influenced by the environment of different species that they live in. For example, if you have free-living organisms that are living on their own, so they're not living with that much with other organisms, you have several hundreds of such enzymatic reactions. Why is that the case? Any thought on that? Why do we have more reactions in free-living organisms? Wrong answers are welcome again, so please. Well, free-living, they're living on their own. So why do they need a lot of reactions?
They can't really split the work with anybody else, any other cell.
Yeah, that's true. So they cannot actually take advantage of other people's, well, this is not a nice way to put it, but wastes, right? Or efforts or services. So if you're living on your own in a forest, so you actually need a bunch of other things, compared to what you have at home. Yeah, so this is an example of a metabolic network. Often when you see these networks, they look really complex, and sometimes they look like subway maps and so on and so forth. So I'll show, and I'll get to that slide in a bit. But usually these are the blueprints of all the reactions that cells need in order to survive. So these are basically life-sustaining reactions that we need to survive. Another database that is really interesting and is really good to look at biological networks, especially metabolic network, is the Keck database. So, well, this is a huge database. There are so many modules in there. There are so many databases that you can browse. But what I would like you to do is to go to Keck Pathway database and just search it in your browser and then get to Pathway database. And what I would like you to do is to search for folate or folic acid. We call it folate biosynthetic pathway. And we want to compare this network in human and E. coli. Have you been able to get into it? Perfect. I see that people are looking with the phones. That is great. One nice thing about these databases is that you can actually browse it on your phone. So that's really nice. And yes, so we have this metabolic pathway for folate biosynthesis. You might have heard of folate. It's folic acid, vitamin B9, basically. And what's really interesting here is that you have, of course, a complex structure, a bunch of arrows and a bunch of points here or nodes. So, but what's really interesting here is that so these nodes and these the circles that you see are metabolites. So these are molecules that we need for survival. The arrows in this graph show the reactions that get one metabolite to another or convert one to another. And this is happening by the aid or by the enzymes that we have in the cell. That's why you see these boxes. So these are actually the enzymes, and these numbers are the chemical reactions that these enzymes perform. Why do we have like a code in here? So the reason is that there are so many enzymes, so we actually invented a system to be able to say, what's, for example, 2.7775. It's a catabolic reaction that does X and Y. And these are actually the sort of the names that we use or the IDs that we use for enzymes. So the next thing to do is now compare this network for human and E. coli. So let's do this. So what we can do here is that you can select or browse to the organisms and choose human and choose E. coli, and then we get into our discussion after that. So the first thing that you see, I guess you have been able to do that. So the first thing that you see is the presence of green and, blank boxes, right? So you see that, right? That's perfect. So what it means is that some of these enzymes exist in one organism, but they do not exist in another. So whenever we have a green box, it means that we have that enzymes, let's say, if this is for E. coli, we have that enzymes in E. coli, right? So these are all the enzymes that we have, all the green boxes here. But in human, that's actually a good question for us. What's the difference between these two? What's really apparently clear if you look at these two graphs? These are like those questions you get that, find 5 differences between. But, one point is that everything is similar, right? The backbone is similar. The only thing that is different is what? Yeah, go ahead.
Maybe the cluster on the right.
Okay, that's interesting. You mean this one, right?
Yeah.
So what you mean is the colors or the presence of this cluster? Oh, good. That's it. Well, that's true. If you mean actually that in some cases we have basically more colorful boxes or a cluster of reactions, and in other cases we don't have, that's actually the right answer. What's really interesting is that folate comes here, so this is folate. Now I would like to think about this part of the graph. So what is the difference between human and E. coli, right, in this part of the graph, if you compare this with that? Look at green boxes. The rest is, the structure is the same. So somebody gives the wrong answer, just, so they look completely the same. Yeah, go ahead, please.
So for human, the circle or the pathway on the right is much stronger than for equal.
Much stronger. What do you mean by the strength? So what's the point? What's the meaning of a strong here?
Probably just more, it more uses that pathway.
Yes, that's true. So what you mean is that you have more green boxes, right? More green boxes for E. coli. So you see they're all boxes here, they're all green here. So this is actually more or less a perfect path or route. And then somehow for human, it gets broken somehow here, so we don't have anything here. And that's a very important difference. So what it means is that bugs, like bacteria, E. coli in principle, and other bacteria would have actually pathways to synthesize folate de novo, meaning that they actually have the ability to synthesize folate, because you see there are so many reactions here. They could actually use inputs, different metabolites, and then get all the way on all the reactions done to get to produce some folate. But in human and some higher organisms, we don't have that luxury, right? So we have actually reactions or processes around folate, but you see this is an island, so this is pretty isolated. That's why we need to take folate and folic acid from diet, right? So that's why we need to, well, in principle, eat a lot of leafy vegetables and so on to get folate, or take supplements. And the reason is that E. coli is able actually to synthesize folate de novo. But that actually brings or sort of shows a very interesting question that I'd like to ask. Before that, is there anything unclear here? So we know E. coli is a bacteria, so it's a bacterium, and in principle, we can be infected, right, by E. coli. So we can actually, this is a pathogen, right? There are so many sort of prokaryotic pathogens that might exist, right? And then if this structure is the same, it means, well, let me ask the question differently, since we wait for this. Okay, so the question is that we have a bacterium here and humans here. If you want to kill the bacterium inside human cells, where do you attack in principle? And your aim is really to cut the growth. So one key point is that folate is very important for growth. So it's something that if you don't have, you end up not growing, actually. It's pretty important. And if you cut folate, right, so you actually can lower the growth of bacterium inside the cell. Is it something doable, or is it something, possible? Any thought on that? Just based on these two networks that we have. Go ahead.
We could target pathways that are not present in humans, so from the brain part you highlighted.
You mean we kind of try to attack, kind of cut this, right? Yeah, that's correct, absolutely correct. So what's really interesting is that we can cut this. The reason is that, well, we can take folate from nutrients, from diet, but the poor E. coli needs to synthesize folate. So if we cut it, there's no way actually that they can take it. And this is exactly the kind of, a great part of antibiotics actually work like that. So they would actually attack this step. So sulfonamides, are actually types of those drugs that would really cut it, because we can actually leave, but those bugs can't. And this is really interesting. So it's just just by looking and by browsing these networks, we can get ideas on how to approach a sort of medical problem that is interesting in medicine. One thing that we know you might actually come across, if you look at literature about metabolic networks, is that people often show similarities between metabolic networks. and metro maps or subway maps. So here you have a metabolic network in human, and then here you have the subway map of New York. So, well, an interesting question for us is what are the similarities between these two maps? So what's really interesting that you see that is really common between these two maps? Is it similar? Kind of. Huh? So So we won't move forward until one person would give a wrong answer. Oh, go ahead, please.
Yeah, so we have those regions that are not really connected to many things, and then we have those hubs that are connected to a lot of things.
Perfect answer. This is the perfect answer. So we have hubs in both networks that what would hubs represent? So these are actually the regions that are highly connected. Probably those are the regions that are really important. So you can think of some avenues that people want to visit or whatever, or reactions that are of central importance for metabolism. We have also detours, right? So we have also different ways to get and navigate through these networks. And well, and that's actually very important because if something happens, let's say if your aim is to get from this point to this point, well, of course, you see, this is a very sparse part of the network. But as long as you get here, so there are two ways to navigate it. Like in Zurich, I can come to Milchsburg from different places, basically. And that is one of the concepts that network can offer and can help us to understand, which we call robustness. So networks could be robust because if you somehow mess with a node, there are ways to circumvent and there are ways to really get to other destinations.